Hi guys, today we are going to talk about DNA and DNA replication. DNA is one of my favorite topics and you really could spend weeks and weeks on it, but we're just going to cram it into one unit. All right, so let's go ahead and get started. So the history of DNA. So there's a lot of different players that are involved in the discovery of DNA, whether it's actual DNA or it's the structure of DNA. And so I'm just going to briefly go through these. So in 1902, Walter Sutton discovered DNA in chromosomes. Frederick Griffith, 1928, he's going to work with Streptococcus pneumonia, and he is going to conduct transformation experiments. This is really important because it's going to designate what is hereditary material. Then we have Levine in the 1920s. He's going to determine 3 parts of a nucleotide. You guys are going to know what that is. We have Hershey and Chase. They are going to use bacteriophage or viruses to show that DNA, not protein, is actually what is inherited. Then we get into kind of the big players in the structure of DNA. So Rosalind Franklin, she is going to be credited with using X-rays to photograph DNA. We have Chargaff. He is going to determine that the role of adenine is equal to that of thymine, or A and T. And cytosine and guanine are also equal. And this is called Chargaff's rule, and this will make sense a little bit later. And then we have Watson and Crick. So Watson and Crick are going to be credited with discovering the structure of DNA, which is a double helix. But later on in the unit, you are going to watch a video called The Secret of Photo 51. And this is going to talk about the controversy behind the discovery of the structure of DNA and how Rosalind and Franklin and Watson and Crick are all involved in that. All right, so what does DNA look like? So DNA is going to have a sugar phosphate backbone. It's going to alternate. So this is kind of a cartoon picture of DNA, but it's really good because it simplifies it, okay? So you have the D, which is your sugar. In the case of DNA, it's deoxyribose, and that plays into the name DNA because DNA does stand for deoxyribonucleic acid. So that deoxyribose comes from the sugar. And then we have a phosphate. Sugar, phosphate, sugar, phosphate. It's going to alternate. It's going to be the backbone of the ladder, as I like to call it. Then we have 4 chemical bases, and I talked about those a little bit earlier. So we have thymine, that's going to pair with adenine. Guanine is going to pair with cytosine. Okay, so you can see over here, thymine pairs with adenine, and guanine pairs with cytosine. Okay, A always with T, T always with A, C always with G, and G always with C. And you can also see down here we have bonds, and these are going to be hydrogen bonds. Okay, so if we're comparing DNA to a ladder, the sides of our ladder are going to be alternating sugars and phosphates, and then the rungs or the steps of our ladder are going to be those 4 chemical bases that are held together by hydrogen bonds. So what are those bases again? So 4 chemical bases. We have adenine, thymine, cytosine, and guanine. Again, you guys, A always pairs with T, C always pairs with G, and vice versa. And they're held together by hydrogen bonds. Now, hydrogen bonds are weak bonds, and we will talk about why this is important later. The bonds that hold our sugar phosphate backbone together are extremely strong. They're difficult to break down. So you can kind of see the pairing. So what does DNA do? So DNA is the genetic code. So any living organism, whether it's bacteria or humans or an elephant or even plants like strawberry plants or corn plants, they all have DNA, and they all have that basic structure of DNA with the 4 bases and that backbone. And that's why we say that DNA is universal. Okay, and so although the combination of our DNA is going to be different between those species, the way that it's written out, if you will, it's different. DNA is the common language between all living organisms. So DNA is important because it's that major instruction manual that tells our bodies how they're going to be. So from our physical characteristics, so what we see in outwardly appearance, and also what we don't see. So things like what we might be allergic to, or what are diseases or genetic disorders we might be susceptible to. All of that information is contained within our DNA. So our DNA is going to code for 20 amino acids. These amino acids will then form proteins, and those proteins are really the building blocks of life. Okay, important. Remember of that. So as we go through this unit, we'll actually talk about the central dogma, which is DNA to RNA to proteins. So that although DNA contains all of the information that is needed, proteins, which are built out of these amino acids, are really the ones that are doing the work. They're the workers. So DNA replication is the process in which DNA within a cell makes an exact copy of itself. So why do we need DNA to replicate? We talked a little bit about this during mitosis, and the reason why we need it to happen is because when cells go through mitosis or cell division, that those two new daughter cells have a complete set of information. Now, whether or not all of that information is used, okay, most likely isn't going to be, it does need to be contained within that cell. Question number 2, so during which phase of the cell cycle does DNA replicate? So we also talked about this during mitosis, but it occurs during the S phase, okay? So remember, this is the cell cycle, the cell's life, as I like to call it. We have our interphase, which contains G1, S, and G2, and then it goes into mitosis and cytokinesis. So during the S phase, the synthesis phase of DNA, or excuse me, of the cell cycle is when DNA is replicated. Now, there's different models that were thrown around throughout history. So up here, you can kind of see we have our original DNA, and we'll go through each one, and then I'll tell you which one is the one that we go through. So we have conservative replication. This is when the original strands are conserved, and then our new strands are in a completely new model. Then we have dispersive, where little pieces are cut up of our original, and then the blue is all new DNA material. So they're all kind of put together like a jigsaw puzzle. And then the last one is semi-conservative replication, and this just means that half of our strand is new and half of our strand is old in this, and then half of our strand is old and then half of our strand is new. This is semi-conservative replication. So again, conservative just means that the original strand's left intact, and then the copy is a completely new copy. It doesn't contain any old material from the original strand. Dispersive, again, I like to think of it like a jigsaw puzzle. We cut it all up and then put it all together. So we have different combinations of new and old. And then the last is going to be semi-conservative, and that's going to produce a DNA molecule that has one old strand and one new strand. And then in this, we have one old strand and one new strand. And our cells actually go through semi-conservative replication. So what happens, or what is going on when DNA is being replicated? Something really important to understand too is that DNA never leaves the nucleus. So when DNA is being copied during replication, it occurs inside of the nucleus. It never leaves the nucleus. All right, so let's talk about the process of DNA replication. So this is a pretty simplified explanation of it. If you take another class later in college, you'll probably go into more detail, but this gives you a good basis of what's going on during DNA replication and some molecules that are needed for it to carry out. So again, DNA replication is that process where an entire double-stranded DNA is copied, and it's going to produce an identical double helix, okay? So that when a cell goes through mitosis, each cell has a complete set of DNA. So there's a few things that are going on. So first thing that's going to happen is helicase. Now helicase is a molecule that's going to unwind and unzip the DNA. So it's actually going to break those hydrogen bonds. Now earlier I said, well, why do those bonds need to be pretty weak? Well, this is the reason why. Okay, it needs to be weak so that this helicase can actually break them easily. So then it's going to then start at our replication bubble, okay? These also can be called replication forks. You can kind of see they look like a fork. And this is our bubble right here. So these are our replication forks. And this overall thing is our bubble. So again, the two strands are going to separate as the hydrogen bonds between those base pairs are broken. Then we have our DNA that is going to form those replication forks. Now what's happening? So helicase is going to completely unwind that DNA strand. Now these bases want to come back. They have an attraction for each other. And so now we have another molecule, it's called SSB, or single strand binding protein. And this is just going to keep the strands from coming back together or re-annealing while DNA replication is going on. So now we need a primase, okay? So just like you need a primer before you paint a wall, that's what we need in DNA replication. So we have primase that's going to come in and it's going to make an RNA primer. Now these primers are going to tell the DNA polymerase where to start. The DNA polymerase is responsible for bringing these bases to the strands. So you're going to have your leading strand, okay, and this is going to be continuous, okay, so it's going to continue down with no breaks. Then you have your lagging strand, and the lagging strand takes a little bit more work. And so what happens is the DNA polymerase will move down until it hits that fork, and then it can't go any further. So then it has to go back here and go. And so when it does this, it actually creates fragments. Okay, so whereas our leading strand creates one continuous strand, our lagging strand does not. So again, DNA polymerase is going to start on the leading strand, it's going to continuously make our DNA. Then our lagging strand is only going to create them or create these fragments discontinuously. So you'll see in this image, the top or the leading strand is completely done, okay? But then on the bottom, we have these discontinuous pieces, and these are called Okazaki fragments. So we have to go back and join them together, okay? But before we do that, DNA polymerase is going to remove those primers because this is DNA, not RNA. We're going to replace them with DNA. I'll go back. So just replacing those RNA primers. Then ligase is going to come in. So ligase is going to come in and it's going to bond the backbone together so that we have a continuous strand, and now we have two identical double helices of DNA. I'm going to show you guys this short video. Hopefully it plays for you. If it doesn't play for you while I'm giving notes, you guys can go into the notes. I've linked them, and make sure and watch it because it just gives you a really simplified explanation of DNA replication. All right, so that is DNA and DNA replication. If you guys have any questions, make sure you email me, Zoom me, or Skype me. Thanks, guys.